Welcome, everybody, to the Hassenfeld Institute Seminar today. I'm so happy you're able to join us. My name is Amy Anthony. I'm the Communications and Outreach Manager for the Hassenfeld Institute. And I'm pleased to introduce to you Dr. Eric Morrow, who is the co-lead of the Hassenfeld Institute's Autism Initiative, and he's going to get us started. Dr. Morrow.
Hi, everyone. So it's my pleasure to introduce, on behalf of myself and Steve Scheinkoff, Steve's there, who's also the co-lead for the Autism Initiative within the Hassenfeld Institute, a special seminar today by James McPartland from Yale Child Study Center. We're really pleased, Jamie, both for Steve and I and the group in the Autism Initiative that you could join us today. James McPartland is a professor of child psychiatry and psychology at the Yale Child Study Center. He's been there for his full academic career from the point of being an intern. Did his PhD at University of Washington with the preeminent autism psychologist Geraldine Dawson, where Jamie studied electrophysiology measures in preschool children. He then went to Yale, where he did his internship and postdoc. His entire research career has really been devoted to neurodevelopment and autism, which reflects just such a true depth of knowledge. The work that he added to his research repertoire when he was a postdoc, and then ultimately when he started his own lab, he added to his initial studies in electrophysiology, but he added eye tracking studies and face perception studies, and really branched out into neural underpinnings of autism, including social reward. He's now the director of the PI on a very large national, very interesting public-private partnership called the Autism Biomarkers Consortium, which I think will be a big part of his talk today. He is at the Yale Child Study Center, the associate director of an electrophysiology research lab. He's also a licensed clinical psychologist who spent much of his career not just doing research, but also doing clinical work. And at Yale, he directs the Developmental Disabilities Clinic, where he's also been quite expert in autism diagnosis and integrating research advances into the clinical setting. He's numerous honors. He's chair of the Autism Science Foundation Scientific Advisory Board. He's an associate editor at the Journal of Autism and Developmental Disabilities. He's a fellow in the American Psychology Society, and has won prestigious awards, including from Brain and Behavioral Foundation, where he won the Claremont Prize. We're really thrilled to have you visit us at Brown and to give a seminar. It's really an honor for us to have you, and we're really looking forward to your talk. One quick thing, so Jamie will, the format for today with Zoom, Jamie will present his seminar, and in the end, he'll leave time for questions, and we'll hopefully have time for a nice short discussion at the end. But we'll recommend that you put your questions in chat, and I'll help Jamie call on each of you to ask questions. And I know there'll be a lot of fun stuff to discuss. So Professor McPartland, we're thrilled that you're here. Thank you so much.
Great. Thank you very much, Eric, for the very kind introduction. It's fun to have the chance to talk with you all today. And I appreciate the invitation. And I'm going to start by, what I'm going to start and finish by is talking about really what has led up to the Autism Biomarkers Consortium for clinical trials and the way that we're using the study to try to advance biomarker qualification with the FDA. These are the groups that support my research and support me, and I don't think there's any conflict with the things that I will talk about today. here's a broad roadmap of what I'd like to discuss. So I want to start by talking a little bit about where we stand with biomarker research in autism, what I see as some of the key objectives for biomarkers in autism, why they're useful. I want to highlight some of the things that I think of as scientifically important in evaluating biomarkers and also as practically important. And then I'll give an example. I'll tell a story about a biomarker and progress in understanding some of these things. And I'll describe some of the reasons why I feel like we haven't made it as far as we could. And then I'll describe ways I think that we could make it a little bit farther in terms of pursuing more rigorous research studies, and then also by using biomarkers maybe in more creative ways to translate them directly to care. And as a start, I want to review some of the tools that I am able to exploit as a scientist. So things that we do in my lab or in collaborators' labs, things like EEG, positron emission tomography, functional near-infrared spectroscopy, eye tracking. So what an incredible armamentarium of really powerful instrument to develop insights into what's happening kind of neuroscientifically in autism. And as Eric mentioned, in addition to doing science, I also do clinical work. And I want to contrast this array of tools with the tools that I have at my disposal when I go to the clinic. And so when I go to the clinic, this is my tool. I have my clinical eyes. I have the same tool that was being applied in the 1940s when the first description of autism were written. And we've really gotten very far, considering that this is what we rely on. We can use it in sophisticated ways. We've built beautiful, reliable scaffolds for clinical insight and clinical judgment. But I believe that there are inherent limitations on what we can do based on clinical judgment alone. And I think that the way to move forward from this is to find biomarkers that can effectively, validly, reliably be applied in autism. When I say biomarker, I think biomarker can be understood in different ways. I actually think in autism, biomarkers are often misunderstood. This is one definition of biomarkers. This is taken from a working group document, a group of people that came together through the FDA and NIH to think about how biomarkers would be applied in these kinds of contexts. And they defined a biomarker as a characteristic that's measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. And so, I mean, two takeaways from this definition are that a biomarker can be a lot of things. A biomarker isn't just something that tells us whether a person has a condition or doesn't have a condition. And the other takeaway, I think, is that really what is a biomarker? It's an objective measurement, right? This doesn't say that it has to be in your brain. It doesn't say that it has to be in your genes. It's something that it's objective, that tells us something about an aspect of a disease or condition. When we think about how we apply biomarkers to autism, this same document classifies seven different categories of biomarker, and I'm going to mention 3 different categories, but we could think about different purposes. We might have a biomarker that is prognostic, that understanding a person's values on this biomarker tell us something about that person's course, the developmental course. We might have a pharmacodynamic or a response biomarker, something that tells us whether a treatment, whether it's a behavioral treatment or a psychopharmacological treatment, is engaging a desired target in the brain or elsewhere. And then we could also have a third kind of biomarker. And I think this, in autism, I think that we too, a fault, tend to focus on this kind of biomarker and then a diagnostic biomarker. And when I say to a fault, I think because most people, when they think biomarker and they think autism, they think of this as a biomarker that is diagnostic of the condition, that this is a biomarker that tells us who has autism, who doesn't. And I think that that's a tall order for autism, in that autism is not a thing. It's not a biological entity. This is a figure taken from from a paper by Eva Loth and colleagues in the EU Ames Consortium. And implicit here is that there's many, many, many different kinds of people, kinds of etiologies that go into autism. So maybe, and there's excellent scientists pursuing the idea of a diagnostic biomarker in this way. I'm going to really think today about a different kind of diagnostic biomarker, one that I would say is diagnostic of a subgroup of people with autism. So in this really heterogeneous class, can we find biomarkers that help us parse into more meaningful subgroups? I mentioned, a moment ago about some of the things that I see of as shortcomings for our clinical abilities, our clinical judgments in autism. And this is a prime example. So we actually, in 2013, with the DSM-5, we discarded the subgroups that we had developed based on clinical judgment because we didn't find them to be reliable. That doesn't mean that there aren't subgroups that could help us understand who's going to benefit from what treatments, who's going to respond to certain kinds of treatments, different biologies. There's many, many different potential uses of understanding these kind of subgroups. Another way, I call it diagnostic really because that is the particular taxonomical term that the FDA uses. Another way to think about it would be as stratification markers, so biomarkers of stratification. These are now, moving away from what the FDA thinks to the kinds of things that I want to understand about biomarkers in autism, or things that I think would be important. So one thing that I think would be important is that a biomarker is sensitive to diagnostic status. I don't mean to sound contradictory. I don't mean that it defines diagnostic status, but I do think if we're not seeing biomarkers that in some way, at least on average, don't look different between people with autism and people who don't have autism, then it might be low odds that biomarker is telling us something meaningful about the autism. We would want biomarkers that would associate with symptoms. And I think this is important. I think about it for those of you familiar with RDoC or the research domain criteria, I don't know that we need to be looking for biomarkers that are autism biomarkers specifically. We might be looking for biomarkers that tell us something about a functional process, be it face perception, social motivation, speech comprehension, auditory processing, what have you, that would be relevant to autism, that I would call an autism biomarker because they could be applied to autism, but are associating with symptoms rather than just the diagnosis itself. To flip that idea on its head, we would also want biomarkers to be associated with symptoms in functionally specific ways. And perhaps this is best defined by an example. And if we had a biomarker, for example, that we thought was telling us something about social ability, we would want to understand that it's not telling us something specifically about intellectual disability or intellectual ability. And that would be hard to know because intellectual ability to an extent is going to cap social development. But if we effectively applied an intervention that improved social function and we had a biomarker that we thought was measuring social ability, was actually measuring intellectual disability, it might not move. And so I think understanding what they measure and what they don't measure is important. We don't need, it's important to understand in autism whether biomarkers are applicable across development. When we talk about autism, we could be talking about a person who's 3, a person who's 30, a person who's 60, and it's not always clear whether the same kind of measurements can effectively be applied at different points in development, and that's something that is understandable but needs to be understood. Depending on the kind of biomarker, we want biomarkers that are robust to variation in behavior. Again, I'll give an example. This wouldn't matter necessarily for a genetic biomarker. A child's behavior during a blood draw is not going to affect the content of the genes. Conversely, when we're doing an EEG, which I'll talk a lot about today, a child's behavior during the EEG could actually provide much, much, much more information in our EEG recording than what we're trying to measure, say a visual response to a face. And so understanding the influence of behavior and understanding that variations in behavior during data acquisition aren't the source of the signal or don't obscure the signal is really important in autism. We would like biomarkers for certain purposes. And I should, I wish I'd said it a moment ago, I didn't. When we talk about, like stratification, prognosis, another way to describe the purpose of a biomarker, the FDA term is context of use. And I want to say that now because I'm sure that I will slip into it. And so for certain contexts of use, we would want to see that biomarkers change as clinical status changes, as a person gets better in treatment, biomarker values become less extreme. And then I also like to consider aspects of biomarkers that are practical aspects, because really the goal of some of the work that we do is to find biomarkers that could be applied at scale in large clinical trials. And ideally, right, the dream is that someday some of these biomarkers could shape clinical practice. And there are certain things that will govern how useful they could ever be in clinical practice. And so one is whether a biomarker can be used in populations with special needs, or pediatric populations. For example, the slide I showed earlier showed me lying in a PET scanner, right? This is a tool that we can use to understand autism, but is due to its cost and inaccessibility, is going to be something that's hard to use. And because you can't expose children to radiation most of the time. And so that's an example of something that wouldn't be so viable. It's also an example of something that's cost prohibitive. If we wanted a biomarker that we could use at scale, we would want it to be relatively cheap. And then really to finish beating up on PET, which I actually think is awesome, is it's not accessible, right? We would want biomarkers that are easily accessed. And just as an illustration, if let's say we had a biomarker that was awesome, but it was could only be deployed in Autism Center of Excellence, an NIH Autism Center of Excellence, this would be its reach. If we had a biomarker that could be deployed in any hospital, this would be its reach. And obviously, this is what we'd like to shoot for when we think about utility, ultimately, of some of these biomarkers. I'll talk today a lot about EEG biomarkers. So EEG stands for electroencephalogram. It's a method of recording brain activity. It measures the direct firing of masses of neurons recorded directly from the scalp using a net like you can see here. So this is an elastic mesh with small, soft rubber pedestals embedded in it and a sponge inside each one. By dipping the whole net in salt water, these sponges absorb the salt water and become conductive. And then when they're placed against a person's scalp, they can pick up the electrical signal and can see the firing of neurons. Typically, EEG is done in two ways: at rest, to look kind of the oscillatory idle of a person's brain, or in response to perceptual events or tasks. So to try to specify the electrical signal that reflects a particular cognitive process that you're inducing via things that you make happen in a person's environment. Some nice things about EEG for the purposes that we'll talk about today are that it works. In people with autism, it works. In children, it works in people across a range of cognitive abilities. It's viable. It's a non-invasive technology. There's many ways we can acquire EEG. The nets that we use, one of the things we like about them is it's applied in one fell swoop, and we don't have to abrade a scalp. And so it's pretty user-friendly. Really, it's like wearing a tight, damp hat. And so a lot of people can tolerate it, and it can tolerate movement. So when a person moves around in an EEG, which children do, it doesn't ruin a recording. You'll lose data during the trials on which they're moving, but it's pretty user-friendly technology for children and individuals with neurodevelopmental conditions. It's also a pretty practical technology. So when we think about EEG, it's expensive to acquire an EEG machine, but when you have one, the cost of acquiring data is salt water and latex gloves. And so it's not expensive. And EEG is also accessible. When we think about EEG, not this system that I've pictured, but there's an EEG in just about every hospital in this country. It's already used at the population level for screening, for seizures, for hearing problems in newborns. And so this is a technology that, should a discovery be made, it could be implemented at scale in feasible ways. And then one last benefit for autism is that EEG is something that has been applied extensively and fruitfully to understand social communicative development in typically developing populations. And so we have a nice kind of solid foundation of understanding to try to understand development in people with autism or atypical social communicative development. And here is a particular example of EEG. This is what we would call an ERP, an event-related potential. And so this is a signal induced specifically by presenting a person with human faces. And so this tells us the neural response to faces. And what I want you to pay attention to here are the amplitude. So this is microvolts on the y-axis, and the x-axis is timing in milliseconds. And so you see this negative deflection highlighted in purple is called an N170. And what that means, it's a negative going component, the N happening around 170 milliseconds. And it's a very, very well-studied component in typical development. It represents the recognizing a face as such. So one of the earliest face-specific stage of processing. And this blue line that you see here is what an N170 looks like in a group of typically developing adolescents and adults. And these are data from the first study that I actually did as a graduate student in 2004. This is what it looks like in a person with autism. And so what we see is that they do show an N170. What's significant and what's different from typically developing people is the timing of it. So people with autism show a longer latency, a slower brain response to faces. Now, this was the first study, and I'll talk about how we followed up. We understand a lot more about this phenomenon than we did at the time. And as I go through some of the things that we've come to understand about this N170, I'm going to keep referring back to that list of things that I care about, that kind of list of biomarker attributes that I care about from the start. And so to refer back here, so we're seeing that the N170 latency is slower in autism relative to typical development. It's sensitive to diagnostic status. As part of the same study, we also looked at how well people recognized faces. And we found that people with autism made a lot more errors, almost twice as many errors on a standard neuropsychological test of face recognition. And we saw that the number of errors they made was associated with their N170 latency. And so again, if we think back to our list, we're seeing that this biomarker, the N170 latency, also shows a relationship with symptomatology. One of the next things we wanted to understand, and if you recall my list, we talked about whether it's telling us something that really specific about a cognitive process, something generic. And we can think about that in terms of face processing in autism. If we see a delay for faces at the N170 in people with autism, is it telling us that they have a slower response to social information, or is it just telling us that they have a slower response to visual information? And this is it was a tricky thing to study. We got an idea from a child that I was working with at the time. Let me show you a really quick video, and let me just double check that my sound is shared, and it is.
These W's look like they're on top of each other.
They sure do. You a picture?
A picture?
Yeah.
WPS?
Oh, that's just another.
Hey, that boy's not underwater.
He's got a mask and a snorkel.
And it looks just like a J.
You're right, it does look like a J. So this is a boy, obviously. This is a 3.5-year-old boy on the spectrum who's doing very well. And you can see that he's really interested in letters. And this is a common interest in people with autism, despite difficulties with communication being a prominent feature of autism. Many people with autism, most people with autism, learn to read, decode, and recognize letters as you would expect based on their overall intellectual ability. So we had an idea for a study, is that maybe we could look at brain response, take a look at the way we had to faces, and then contrast it with letters, because it turns out you actually get a differently lateralized, a left lateralized N170 to letters when you learn to read an alphabet, any alphabet. And so what we did is we compared their brain response to letters of the alphabet, of our Roman alphabet, and we compared that to letters of a made-up alphabet, of pseudo letters. And then we compared faces to house. And so basically the way this works is if what we think is happening, this is telling us something specific about social information processing, we should see some differences in people with autism here, but not here. Conversely, if this is just a generic problem with specializing for a percept or a generic delay in kind of visual processing, then we would see difficulties in both areas. So let me tell you what we saw in both of these domains. Beginning, and also this gave us, when we designed the study, we did in a younger cohort so that we could see if some of the differences that we'd seen in the older sample were also true in a younger sample. And so for the social domain, we saw the kinds of things that we had seen before. We saw poor face recognition in people with autism, and we saw that they showed a slower N170 response in their hemisphere compared to age and IQ match typically developing children. And so we're seeing that the delay that we saw in the adolescents and adults is also evident in children, which is cool. Developmentally, we're seeing the same effect across a large age range. When we looked at the non-social domain, we saw a few things. So behaviorally, we didn't see any kind of deficits. People with autism showed comparable word reading and comparable phoneme decoding to the typical participants, and everybody showed scores in the average range on the standardized test. And when we looked at their brain response to letters versus pseudo letters, you can see here letters in purple and pseudo letters in green. What we see is that everyone is showing the expected specialization in terms of enhanced amplitude to letters relative to pseudo letters. And this is amplitude, not latency. But when we looked at latency, we didn't see any differences in latency between the groups. People with autism were as efficient as they were. The only difference that we saw was that when we think about letters typically being left lateralized and faces being right lateralized, people with autism showed a stronger, actually more recruitment of right hemisphere than the typically developing individuals. And so if we think back, so what this would indicate to us, this pattern of results, is that the N170 delays that we're seeing are not just generic for any kind of visual stimulus, they are specific to faces. And we therefore infer that they're telling us something specific about the social domain. The next study that we did to try to understand how the N170 works in autism focused on visual attention to faces. So there's a few things that are important to remember. The N170 latency that we've talked about is actually modulated by gaze. And when you direct a person's visual attention to the eyes of the face, you see a faster N170. We also know from many, many eye tracking studies, people with autism tend, and clinical accounts, right, that people with autism tend to look less to the eyes of the face. So therefore, could it be possible that these N170 delays that we're seeing are not really telling us something different about, the way the brain is responding to visual information, but is telling us that different visual information is taken in? That maybe people with autism are showing a slower N170 because they're not looking at the eyes of the face. And so what we did is controlled for it. And so we had a crosshair appear on screen preceding the face. So these are not veridical. This is just to illustrate. And that crosshair then directed attention to the eyes, to the nose, or to the mouth of the subsequently appearing face. And so what we wanted to see is when we're sure everyone is looking to the eyes, do the differences in N170 latency go away? Are the differences we're seeing in autism just an artifact of visual attention? And they're not. And so what you see here is this is a plot of N170 latency. You can see attention to the eyes, attention to the nose, and attention to the mouth. And we can see that when we look at when people are attending to the eyes, we see the typically developing people showing a shorter N170 latency as we would expect, but we see, rather than that abolishing the N170 delays we see in autism, they're most prominent when we're directing attention to the eyes. So overall, people with autism, continue to show a longer latency, and that's looking to the eyes does not make it go away. And so we're seeing these N170 delays aren't just an artifact of gaze behavior, they're robust, at least to this aspect of behavioral variation. The last thing that I'll tell you about that we looked at with the N170, and this is really, really preliminary data. This is a colleague at the Child Study Center, Pam Ventola, is an expert in pivotal response treatment. This is a behavioral intervention that is designed to improve social function in people with autism. It's empirically validated. It works reliably across a number of studies. What we did is we looked at N170 response to FACEs before and after Pam had worked with the children. So after this 14-week course of treatment, and the kids get better in treatment, the behavioral scores show that they improve. And what we see is, and these are actually, this chart is showing you individual child data. So there's 7 children. So that's why it's preliminary, very small sample, each represented by a solid line, and the average represented by a dotted line. And this is before therapy, and this is after therapy. And you can see that everyone except for one child is getting faster as they get better in treatment. And so we see that their N170 latency is tracking, in this preliminary sample, is tracking with their clinical change, which is another one of the things that we think are important to understand. And so let's revisit the list of things that I think are critical for biomarkers in autism to do. And again, I want to emphasize, no biomarker needs to do all these things, right? That because this represents utility across many contexts of use, but these are the kinds of things that are relevant to understand. So, and for the N170, we see that it's sensitive to diagnostic status. We see that it associates with symptoms. We see that it's functionally specific. We see that it's applicable across development, robust to variation in behavior. We have, promising data that it will be sensitive to change in clinical status. And then, when we think back to our practical matters, EEG is a technology that's viable in this population, that is cost effective, and that is accessible. And so the N170 has a lot going for it, right? So why aren't we in a position where we can rely on something like this to measure whether a treatment works in a clinical trial or to inform clinical practice? And so there are some very, very significant challenges. I want to highlight that this story, the situation that I've described, where there's lots of strong evidence, but not enough evidence to get us to the place of practical usage, is not just true of the N170. It's true for many other biomarkers. Eye tracking is another notable domain where really useful information and promising biomarkers have been developed. One thing that I see as a key problem for all of these biomarkers is limited reproducibility and poor understanding of why we see the failure to replicate across studies. Listed at the bottom of this slide are all the studies that I am aware of that have looked at the N170 in autism since that first study that we did in 2004. And some of them have replicated our findings, some have not. I'm excited that this study by Kang is a meta-analysis that shows if you take all of them together, on average, that people with autism show face-specific delays in their N170. So this seems to be an emergent truth, but why do we see it not true in every study? Well, one reason could be that's just autism. That autism is a heterogeneous condition. There's probably not, a symptom of autism that is evident in all people with autism. So why would we expect that this biomarker looks different in every person with autism? Maybe we would only expect it to be, to look different in people with autism who have specific difficulties in the domain of face perception. So it could be real. It could be individual differences, and that's a good reason for failure to replicate, because it's something that could be studied and could be understood. There are also problems, to understanding it in this literature for bad reasons. Many of these studies are small, and so could have, studies that found differences could have yielded spurious results. Studies that failed to find differences could have been underpowered, and we can't redo these studies. So that's just something that we're stuck with in the existing literature. And likewise, we don't know how variation in procedures among these studies could have affected results. If you use a crosshair, if you don't, if you use our EEG system or a different EEG system, if you show color faces or black and white faces, these are all things that could change the results. And these are all things that differ among these studies, and we don't understand. And we don't have the knowledge yet to infer how they could have added to this kind of the diversity in results. There are other things that we don't know about biomarkers in autism, really across biomarkers. For most, we don't understand reliability or practice effects. So if we think of a biomarker that we'd like to use in a clinical trial, we want to know how stable it is in a person over that span of time, absent an intervention, and we don't know that for most. Likewise, if we're going to take repeated measurements before and after a treatment, we want to know how the act of taking repeated measurements might change a biomarker value. And then lastly, for all biomarkers, I think, except for one that I can think of, we don't have a normative reference. And that one is head circumference, right? So for head circumference, you can go to the CDC website and you can know exactly where a person with autism stands in terms of their head size relative to typical expectations. But you can't do that for an N170. And you can't do that for most biomarkers with autism. And that means our inference is only as good as the study that we're doing. And so that's another problem. So what do we do with these problems? Well, I think that they're largely addressable through empirical methods. And so what we could do is do studies that are more rigorous, that are designed to account for these kinds of problems. And, when we think about, the title of my talk today includes FDA qualification. I'll talk more about it. But when we think about qualifying things for the FDA, that kind of rigor is also going to be something that's necessary for the FDA to endorse something as a biomarker. So let's talk about what these studies should look like. I think these studies should focus on well-evidenced biomarkers. Obviously innovation is important. It's one of the things that we're specifically evaluated on, right? Every time that we propose, we seek funding for a research study. The problem with always driving towards innovation is that it's at odds to an extent with replication. And so I think sometimes it's useful to take biomarkers that are well studied and study them more in these rigorous ways to get them to a place where we've ruled out some of the doubts, where we've fleshed out the areas of understanding, the gaps in understanding. We want to study them in well-characterized cohorts so that we have the information that's necessary to understand the influence of individual differences on biomarker values. And we want these samples to be large so that we have the statistical power to analyze that information. And we want them to be large, including typically developing people, because we want to have both that frame of reference, but also there's no reason that biomarker variation only has to be meaningful in the clinical range, right? Understanding range across typically developing people could be informative, again, with an RDoc perspective. We would want longitudinal design, and I don't mean longitudinal as in lifespan. I mean longitudinal as in the length of a clinical trial, for example, so that we have a sense of of how stable they are or subject to change they are over the span of time in which a clinical trial might take place. We would want methodological rigor so we can rule out sources of noise, and we'd want to use practical assays so that if things work out, they could be applied in meaningful ways. And these are the set of principles that people other than me had in mind, really, when they drew up an RFA for the Autism Biomarkers Consortium for clinical trials, now a number of years ago. So the ABCCT is a multi-site naturalistic study. I'm happy to share my slides, but I'm not going to go through all the details and some of the things here. It's by naturalistic, I mean it's not a clinical trial. We don't administer an intervention. It was a large study, including 399 children, 280 of whom had autism, in the age range of 6 to 11, with IQ in the mild to moderate range of intellectual disability to well above average. We used a battery of EEG and eye tracking markers, so practical assays, and had a design that was longitudinal in the way that I described. So a baseline, a 6-week, and a 24-week. And we actually also drew blood, although we didn't have scientific aims related to the blood, so it's sitting in a repository. I'll highlight a few things that were unique about the ABCCT study design. Is one, it's the funding mechanism is a U19, which means that it's a cooperative agreement. So it's bringing together scientists from our consortium, but then also scientists from the government, scientists from industry, scientists from regulatory agencies, so that we have a really wide-ranging wide-ranging array of expertise to inform study conduct. We also ran this study with I don't use the word unprecedented loosely, but I really think an unprecedented level of rigor in 3 areas. One, in terms of regulatory, this wasn't a clinical trial, but we ran it as if it were. We ran it according to good clinical practice standards. 2, methodologically, we worried about things like the ambient light in the EEG labs and built custom light meters so that we'd make sure that the baseline light was the same everywhere. And so really a high level of paying attention to those kinds of details. And then statistical, where we really were specific about what we were declaring primary variables and making very specific directional predictions so that there's no ambiguity about what we thought was going to happen and what did or didn't happen. And a last advantage of this study design was that we had the opportunity to harmonize some of our biomarker assays with those being used by a parallel consortium in Europe called the EU AIMS, giving us the chance to replicate intercontinentally. I won't go through all the measures, but we ran the full gamut of clinical measures that we currently rely on for clinical research. This was our EEG and eye tracking battery, 4 EEG measures, 5 eye tracking measures. And as you would suspect, the N170 was in the mix. It was the one that we designated as primary for our EEG battery. I'll show you, we concluded data collection for this first phase of the study in 2019. As I'll tell you in a moment, we've been renewed and we're continuing on, but these are some of the results for the N170 from that first phase of the study, which shore up some of the things that I've told you about already and provided some new kinds of information as well. We see that we're pretty effective at acquiring this data on our participants. So 3/4 of people with autism and almost all of the typically developing participants, as we hypothesized, we see across this large sample a delay in people with autism. We see that it's relatively stable. Our biostatisticians would classify this as adequate stability across 6 weeks, and similar, a little bit lower in autism over 24 weeks. And we saw it was related to the phenotype, specifically related to face memory in terms of concurrent correlation, but then also in a predictive way, in that N170 at baseline predicted 24-week face memory. And so you can see the N170 here, as you've seen, you can see the delay, and then you can see the distribution here. Typically developing people shown in blue, people with autism shown in green, and I'll highlight for you the relevance of thinking about context of use. This is not something that would be a useful diagnostic biomarker, because, for example, if you have an N170 latency of 200, almost equal chance that you're typically developing versus a person with autism, whereas you can see that in this tail of the distribution is almost exclusively populated by people with autism. And I'll talk a little bit more about that in a moment. One of the goals of the ABCCT is to approach the FDA with these biomarkers. The FDA has, the Center for Drug Evaluation and Research has a specific biomarker qualification program. We submitted the N170 and another, and I'm not talking about eye tracking today, but an eye tracking marker called the Ocular Motor Index to the biomarker qualification program. Both were accepted into the biomarker qualification program, which is the first of a complicated and lengthy series of steps, but it's a noteworthy step in that these are the first biomarkers to achieve this endorsement for autism and really for any psychiatric condition. And so this is a milestone for our study, and it's a milestone for the field. The way we've described it is, again, as a biomarker that is diagnostic of a subgroup. Specifically, the argument that this purple swath here, this tail of the distribution, represents a biological homogeneous subgroup that could be used to enrich clinical trials, either kind of as an admission criteria or in terms of a focus of analysis, by reducing heterogeneity to improve our power to understand the effectiveness of a treatment. The way that things are proceeding is really, again, cooperatively. And so we actually have gotten to cooperative agreements funded by the FDA to work on biomarker qualification plans with them. And it's a really, to me, a very fun an exciting process because it's uncharted territory, really, for us and for the FDA to an extent, in that psychiatric biomarkers are not an established, a well-established, entrenched biomarker domain. And so the kinds of things we're trying to understand with our colleagues at the FDA is, what are the best ways to draw cut points, continuous distribution? How do we functionally differentiate this subgroup when our clinical measures, we don't have other measures of biology? Autism is a behaviorally described entity. How do we demonstrate that this group is really different in some way? And how do we make sure that these findings aren't specific to our methods? As I mentioned, we were renewed in July 2020. Our progress has been complicated by the pandemic. The structure of the study is that this phase 2 will actually be 3 studies. A confirmation study that is actually going to be an attempt to replicate. Our statisticians don't like to use the word replicate, so we call it confirm. A confirmation study of that first study. The second study is a follow-up study to follow up at a longer term that original cohort, 2.5 years to 4 years after they were originally enrolled, when they'll be 12 to 16 years old, to let us look at stability over a much longer time, to give us a much longer window of time to understand the relationship between biomarker values and clinical change. And then also to look at longitudinal predictive value. What do biomarker values at time 1, way back when, tell us about a child's functioning now, 2.5 to 4 years later? And then the third study is a feasibility study, really to see whether this biomarker battery is feasible in a smaller cohort of preschool aged children. And so because of the pandemic, many of our sites were shut down for human subjects research for a good portion of this year, but we're actually ramping up to begin the follow-up study. As the first, these studies are being done in sequence, and the follow-up study is starting right now. So we're actually at Yale starting to schedule families right now. And so we're really, really excited to carry this work forward. So that's most of what I want to talk about. I want to close by talking about one different element of biomarker research in our group, in autism, and actually given our collaborators in your group, because Linda Carpenter is centrally involved in this work. But one of the this is if what I've talked about with the ABCCT is applying a high level of rigor to well understood things, this is the opposite. This is trying to apply a high level of innovation to things that we don't entirely understand yet. And so the idea is to, if we think about the way behavioral treatments affect behavior in autism, affect the brain in autism as well, is they tend to target social function. And one of the things we've seen from neuroimaging studies is that change in superior temporal sulcus activity, which is a region that's important to social functioning, a region that's implicated in autism, activity seems to be enhanced after intervention. And so an idea would be, well, what if we could apply TMS directly to the STS? And we are not the only ones to have this idea. In fact, a group that's cited there, NE, has actually published 2 studies looking at this, really clinically oriented studies, and what they've found that it does show some promise to improve social behavior. It does show some potential to reduce restricted and repetitive behaviors. But what we wonder is if we could harness these biomarkers to maybe give us a more efficient and maybe more sensitive indication of what TMS is doing. And so could we use something like an N170 here in an early efficacy way or in a target engagement kind of way to show that if we have these biomarkers that we know are associated with autism, could we show that TMS is moving the needle on them in a way that is more proof of concept? There's no inherent value in changing an N170 latency if it doesn't change symptomatology or behavior, but could it tell us for whom behavior change would follow? And so this is really the idea of some of the work that we're trying to get underway now. And just to show you some exciting preliminary data from actually from typically developing adults, when we stimulate the STS, it moves the needle in the direction that we would expect, even in people who aren't showing kind of clinical values. And so if you look at N170 latency, you see that After intermittent theta burst stimulation of your STS, you see that N170 latency is shorter. And we also see this is a measure of eye tracking, is how much time is spent dwelling on the eyes when we present a person with a face. And you can see that everybody is looking more to the eyes when we, after STS stimulation. Again, so this is changing a biomarker in typically developing adults doesn't matter. But we think that it could matter in people with autism. And so this is work that we're actually hoping we have the opportunity to carry out in the coming years. And I'm going to stop there. I'm glad that I've managed to preserve a few minutes for questions. I do want to highlight I want to highlight the contributions of a few different groups. I want to thank, most importantly, the people with autism, the children, the adults, and their families who go out of their way to wear uncomfortable, wet, tight hats and to help us understand things. I want to thank the many clinicians in the Developmental Disabilities Clinic, because this is where it all starts for us. This is our connection to the community. I want to thank the really stellar team of the Autism Biomarkers Consortium for clinical trials. This is by no means everybody, but this is the leadership of the consortium. And then I want to thank the group of people in my lab who really carry out a lot of the work that I've talked about day to day. And you can see our contact information there. So please feel free to get a hold of us after the talk as well, or if you want the slides. And I'll stop there. And if questions have been asked, I'm happy to try to answer them. Thank you.
Wonderful. Thank you very much, Jamie. Very clear presentation of biomarkers in a complex condition, and the depth of the data and the commitment and focus to the problem is clear. So there are two questions to start us off. The first one's from Steve Scheinkopf. Steve, I'll let you ask your own question.
Great. Jamie, thanks once again for joining us here. Hopefully someday you'll be able to hop on 95 and come up to Brown in person. So the question I had was regard to the group difference in the N170 that you reported, and being driven by that tail of the distribution with prolonged latencies. And this is relating to the criterion for biomarkers having to have be sensitive to diagnostic status. And some differences may be reliable but more subtle. So I'm wondering, how does the magnitude or specificity of a difference play into your thinking about the development of a biomarker and its sort of practical application?
It's a good question, Stephen. And really, I have come to think about them really specific to context of use, right? And so, honestly, you could have a biomarker that's useful in autism and not even different on average from typically developing people if the differences within the group of people with autism are meaningful in some practical way. And so, I actually think less about the magnitude of the group average differences, if I was understanding your question correctly, for what we do, and more whether I can identify something that is, meaningful about, that would be like, for the purpose that the context views that we're dealing now, I'm thinking more about, can we identify something meaningful about that tail within the group of people with autism? And that hasn't been easy. Wouldn't it be awesome if we could just pull them out and their ADOS scores were just different from everybody else's, or even if their eye tracking scores were just different from everybody else's? And it's not that simple. And so we're exploring it. One of the things that we've come to think about too is that, if we see limitations in our clinical measures, but then we're using them to validate our biological measures, there's an inherent problem, right? And especially if you look, so one of the examples, one of the things we thought, what should be the thing we look at in that tail? And we said Vineland social domain, right? Then you open up the Vineland manual. What goes into the Vineland social domain? Interpersonal relationships, that sounds right. Play and leisure, sounds like that could be right? Coping, hey, wait a minute. Emotion regulation, superior temporal sulcus, like N170, maybe not. And so we're also we're actually revisiting a lot of our clinical measures to try to think in a different way, not based on what the publisher called the scale, but what we think the scale is actually measuring, and trying to see if that helps us understand. But it's complicated. We've experienced it complicated. If you all know how to solve the complications, I'm listening.
Great. Okay, so we also have a question from Professor Jim Padbury. So Jim is a pediatrician and a neonatologist and a researcher at Brown. Go ahead, Jim.
So thank you very much. I enjoyed your talk, and the slide that really resonated with me was the slide on the subgroups. So, and it resonates with me because it reflects the way we think of the genetic architecture of complex diseases. They're not due to single variants in single genes, but variants in shared networks and pathways. Everybody doesn't have the same networks and pathways altered, but there are clusters of patients that do. So I'm thinking all these characteristics, these phenotypic characteristics of your autism patients are not independent. They're probably codependent or related to one another. And can you use some machine learning, latent profile analysis, cluster analysis to discern what are the clusters of patients, how many are there, and then how well do they map to the biomarkers? Or you could even use the biomarkers as part of your latent profile analysis. And the dependent variable in all this is the biomarker predicted well by the clusters, or is the treatment responses? So there's my thoughts.
They're great thoughts. And you did ask a question. The question was, the snippet of it was, can you use? And I'll answer that very specific question. I cannot use, but there are people out there who can. And that's one of the thrusts that I didn't mention of this project is also open data. And so the genes are out there. They're actually being sequenced by the Broad Institute through and the Autism Sequencing Consortium through the Biomarkers Consortium collaboration, and all the other data is out there. And so the ways that we've looked at the data so far are really oriented towards hypotheses based on the existing analyses that have been done over the past 20 years, right? So I think that those kinds of clever things could absolutely be done. We haven't done them, and ironically, we're not funded to do them, but we are funded to set people up to do them, and we do.
Premised on the notion that these phenotypic characteristics, either the N170 or the eye tracking or any of the any of the features, as they're called in machine learning, are not independent.
Right. Yeah. I mean, I'm not an expert in machine learning, right? So,
but do you think the phenotypic characteristics are independent?
You mean? Can you help me understand what you mean? Independent?
I'm saying they cluster with one. There are going to be clusters of patients that have certain N170 latency, certain clinical characteristics, and they can be discerned in an unsupervised fashion with latent profile analysis or cluster analysis, and then you can look and see how the biomarkers map under these clusters.
Yes, I do think they are independent. Yes, I think that is a viable approach.
Thank you.
I put myself on mute now. I'm off. Are there any more questions? Anybody want to raise hands or put a question in the chat? All right, so we're at the hour. So again, I really want to thank Professor McPartland for wonderful talk from both me and Steve. Thank you so much for coming, and I want to thank everybody for coming to the seminar and participating in the discussion. It's really exciting, and we'll look forward to and as Steve says, hopefully we'll look forward to interacting more and following along the science in person and at some point soon.
There's a famous musician named McPartland.
Yeah, Marion and Jimmy. I unfortunately no relation, and I don't even independently have musical talent.
All right, I'll look that one up tonight.
Thank you so much for having me, and thanks everybody for being so engaged the last hour of the day. So thank you.
Yeah. All right. Bye now. Thank
Bye,
you.
everybody.
Thanks, everyone. Thank you, Dr. Morrow. Thank you, Dr. McPartland. Thanks, everyone, for joining us.